Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Hello, welcome. Glad to see you could make it. My name is Stuart Ozer. I've been with Microsoft Research for a couple of years, currently working in the SQL-server product group. Over the last 2 years, it's been my pleasure to work in collaboration with Dr. Robin Gattel from University of Texas in Austin, who has built an enormous system over the years to curate and analyze detailed structure of ribosomal RNA and other RNA molecules. And we've worked together to evolve the system to leverage database technology, in particular SQL-server 2005 and a lot of new tools to make the process both more productive and streamlined for accomplishing science, but also to facilitate new discovery. And Robin's going to talk about the significance today of RNA science, about the system that he's built, and the evolution of the system utilizing new Microsoft technologies over the last couple of years. And when Robin's done, I'll spend a little bit of time going into some more technical detail on the architecture, and if we have some time, a little bit of demonstration as well. So with no further ado, Professor Gattel.
Thank you. So first, can you hear me? Okay. A few months ago, in the June issue of The Economist, this was the front cover. And I find this interesting and important because starting as a graduate student in 1977, so I'm starting to feel a little old, studying RNA was not considered to be fashionable. It was proteins. But my PhD advisor, Harry Noller, had said RNA is more important. But most people just don't know it at the time. And so I will briefly talk about now how there's a major paradigm shift going on right now in biology, and it's essentially focusing on RNA. So our perspective of how the cell works is going to be changing in the next few years. That's a quote you could. And so I was pleased to see that The Economist was picking up on this. Okay. I am trained as a biologist. I've never taken a computer science course in my life. Some people would like that, others don't. My enjoyment is working with the sequences. Being a scientist, being a biologist, asking specific biological questions. My PhD thesis was to predict the secondary structure of a molecule called ribosomal RNA, because in the lab that I worked in, the first RNA was determined, it was 1,500 nucleotides in length. And I learned very quickly that it was not a simple problem to solve. And so what I started doing at the very beginning was starting to work with computer scientists to help develop new programs. And so what we have here is that I am now collaborating with Stuart and others here at Microsoft to essentially enhance and make a significantly better version of a system that I had been working on for about 20 years. Okay. Importance RNA science. So there's about 5 major sections in my talk. So one is importance of RNA. This is an outline of what's to follow. Grand challenges in biology and the use of comparative analysis. Briefly, not knowing the audience, these are 2 different pictures of a cell, and there's a ribosome, which is a major focus for us, and the ribosome is where you translate a RNA into a protein, and there's all these other components inside the cell. One of the main take-home lessons is that it has always been viewed that the proteins are the most important part of the cell. And RNA was simply a messenger RNA, which is an intermediate between its DNA and the protein that's translated from that messenger RNA. This is the paradigm that I'm talking about is going to change. RNA is going to be involved in many, many stages of what goes on inside the cell, while currently it's thought that proteins are actually regulating and involved in much of the cell's activity. Yes, proteins are very important, but RNA has a lot larger role than it is currently given credit for. This is a figure I found in an excellent book that just came out called Understanding Bioinformatics. It's the best bioinformatics book I've seen. Since I teach a course, they sent it to me, and it's the book I'm going to use. I like this diagram that they show because, just to emphasize a few points quickly, you have essentially nucleic acid world center, and one component here is you've got structure and RNA, and it breaks down into nucleotides and base pairing and RNA structure, and you've got evolution as a major component, and you have translation and as a major component, and you have the regulation as a major component. And traditionally, each one of these are treated as individual dimensions. And many of the components actually within these are treated as an individual component. And one of the things that I've been doing, and a major reason why Stuart and I started to collaborate is, and I will talk about this, is that we wanted to bring multiple dimensions of information all together. Because by doing so, we could then decipher, sniff out more knowledge and information about the system. As previously, each one of these were treated as an individual dimension and was not compared, related with any of these other dimensions. Okay, so what I was saying earlier, previous paradigm, rRNA, tRNA, messenger RNAs are the major RNAs in the cell, and this perspective is changing. And so most of you know about Watson-Crick base pairing, AU and GC, and it's thought that RNA structure is sort of simple, that these will form into a helix. But what we and other people have found is that in reality, you can form all types of base pairing. You could have an AA base pair, a GG, a CC, and that these can form into a three-dimensional structure, and these three-dimensional structures are capable of doing catalysis, which in the past was thought to be just something that proteins do. So RNA, in one sense, is, I like to think of it as a DNA-protein hybrid. It has capabilities of acting like DNA in the sense that it has Watson-Crick base pairing, and it can form a helix, but it also is like a protein in the sense that it forms an interesting three-dimensional structure that can catalyze a reaction. This is one of the major perspectives that's changing. The second one is that it has generally been accepted that the complexity of an organism scales with the amount of protein in the cell. So they have a complete genome of C. elegans, which is just a small worm, and Drosophila, and human, and mouse, and other complete eukaryotes. And they thought that they would find more proteins in these larger, more complex organisms, i.e. humans would have a lot more proteins than C. elegans. What they're finding is that approximately all of these different eukaryotes have approximately 20,000 proteins. But what they are finding is, and this is still sort of an early stage, so there's people that will still debate this issue a little, but I actually believe it's correct, so I'm going to say it, is that it appears that the complexity of the organism scales with the amount of RNA in the cell. So I've now just stated two primary reasons why RNA is more important. So there are several grand challenges in biology. I'm just going to discuss two of them. One of them is to predict an RNA secondary and tertiary structure from its nucleotide sequence. So, example, you start with a tRNA sequence that's about 76 nucleotides in length, and you want to predict its secondary structure, which is its classic cloverleaf, and ultimately you want to fold this up into a 3D structure. A second major grand challenge in biology is to determine the phylogenetic relationships for all the organisms that exist on Earth. And I don't know how many people here have heard of Carl Woese, but in the year I started graduate school, in 1977, he found a third kingdom of life. There used to be a group called the prokaryotes. So you had the prokaryotes, which were bacteria and archaea, and you had the eukaryotes. Carl found that the prokaryotes broke up into two major groups, the bacteria, as he called it, and the archaea. This was based on his analysis of the ribosomal RNA. This is such an important finding. I'm envious. A lot of us are envious. It was published, first page, on the New York Times. These are two grand challenges. You'd also think these two grand challenges are two entirely separate issues. But there's this famous quote, which I like. I think it's true. Nothing in biology makes sense except in the light of evolution, i.e. if we're going to study a biological system, study it from use evolution to help understand the system. So our efforts to predict secondary structure from a single sequence using simple energy rules failed. That's a whole lecture in itself that I won't talk about. Just accept that. So I felt doomed because my PhD advisor said your PhD is to take this 16S rRNA, 1,500 nucleotides long, and predict its secondary structure. And there are something like 10 to the 100th possible secondary structural models for 16S. So I thought, uh-oh. Fortunately, Carl Woese came along and said, no, use comparative analysis to predict the structure. Briefly, what comparative analysis is molecules that do the same thing. For example, tRNA. Even though they will have different sequences, there's three different tRNAs that have three different sequences. They all fold into the same secondary structure, and they fold into the same 3D structure. And down here, we actually compare the sequence identity. So even with only 40% sequence identity in this alignment, you can form the same secondary structure and 3D structure. So our objective is for the 16S and the 23S ribosomal RNA was to find a structure that's common to all the 16S's and 23S RNAs. Okay. Here's a quick overview on its application. Up here in the left is a diagram that has the E. coli sequence for a small region of the 16S shown here as the main sequence, and the actual differences between E. coli and Bacillus brevis shown as those nucleotides that are actually shown here, meaning that the brevis sequence is identical in all the places in which you don't have this second sequence here. And you can predict a structure here, a small secondary structure, that at the time was considered more stable than this structure down here. But this structure down here, you see, you have what's called covariation. So in coli, you'd have a UA base pair right there at the bottom. And in brevis, you had a compensating covariation. The U changed to an A on the left side, and the A changed to a U on the right side. And so 4 of the 6 base pairs in this helix actually had a covariation. So essentially what we set out to do was align sequences. That's what this schematic shows here. And we look for covariation. We look for columns in an alignment that have a similar pattern of sequence variation. And so this A here, this A here, and this A here are 2 columns that actually base pair, and that's shown right here. And you see that in some bacteria, you have some CGs and UAs and GC base pairs, and in the archaea, it's all CGs, 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 and in the mitochondria, you have some GCs and AUs. That's just in the column A, well, I should call it A and A prime, and this applies to columns B on the left and right, and C, et cetera, et cetera. So we here have predicted 5 base pairs, and it's in this structure right here. So this A, these 2 columns, 240 with 286, is right here, and it's CG in E. coli, but as I said, in other organisms, it might be a UA or a GC right here. And the same applies for all the other base pairs here. So again, we are looking for, in an alignment, we're looking for columns that have similar patterns of sequence variation. And I'm just showing a few examples here. We then predicted the entire 16S ribosomal RNA structure as shown here. It's about 1,542 nucleotides in length. And I spent about 20 years working on it, of which at the very beginning we had 2 sequences, and we predicted part of it, and then the 3rd sequence came, and we essentially treated that first structure as a hypothesis and asked, is the 3rd sequence consistent with that structure, or does it add new structure or take out any existing structure? And I kept iterating and iterating and iterating for 20 years. I did other things too. But we came up with this structure, and no one ever thought, or at least I never thought, that we would get a crystal structure. Crystal structure is a high-resolution structure. This is about 2.8 angstroms in the year 2001, meaning I thought that this was going to remain a prediction until I died. It was consistent with many, many experiments in which people would test individual base pairs and make compensating base changes. And if you change or if you change one nucleotide in a base pair but not the other, it might kill the cell. But if you make the compensating base change, it essentially brings the cell back to life. And so we were confident that this structure that was predicted with comparative was correct. But it was ultimately tested once we got the crystal structure. I think it's on the next slide. No. Oh, actually, I'll get to it in a few slides, but I'll say it now. About 97%, 98% of the base pairs that were predicted with our comparative covariation analysis was in the crystal structure.
Ultimately, how many sequences did you have to contribute to that prediction?
Well, that's a slide, an interesting slide that I have in a paper with two sequences that are properly spaced, meaning two sequences that are 99% similar are not going to be very useful here. Two sequences that had about 80% identity, it turned out that about 70% of the structure predicted with just two sequences is in this structure. And as you add more and more sequences, you essentially can keep tuning this secondary structure model. We can still learn a lot more. That's another story in the sense that certain regions will have a special characteristic in organisms just on one branch of the tree. So in one sense, the structure might look a little different on certain branches of a tree. And so we're still not actually done, but we're sufficiently done. Another very, very important dimension is to ask how frequently does a change, a covariation occur? So for example, this 925 base pair, which is shown up here, if you simply looked at nucleotide frequencies, you'd see that about 25% of the sequences are CG, about 57% are GC, and about 82% are AU. It turns out that almost all the GCs are bacteria, all the CGs are archaea, and all the CGs are also, all the GCs are bacteria, and all the CGs are either archaea or in the eukaryotes. What that means is that this event, as we call it, this change, this mutual change occurred once, approximately once in the evolution of this entire molecule. Another example here is this 1357, 1365. We see that there's AGs, GCs, and GAs. In the bacteria, it's predominantly AG and GA. In the archaea and in the eukaryotes, the covariation is actually between Watson-Crick base pairings, GC, UA, CG. And then this third example here is 245, 283. This is where I used to get heckled. Maybe I'll get heckled again. When I actually proposed a CC and a UU base pair. Because all I was looking for was patterns of variation in an alignment. I was not looking for Watson-Crick base pairing. And what this schematic, this diagram is intended to show is that the UU and the CC exchange many times in the archaea and in the bacteria and in the eukaryotes. So the more frequently you get a covariation, or what we would call an event, the more likely it is true, or true in the sense that it's a base pair. So there are actually a number of base pairs that are invariant in the archaea and the bacteria and in the eukaryotes. And if they're invariant, you cannot actually necessarily predict that it's a base pair because it's a chance event that you can form a Watson-Crick base pair. We happen to predict this 925 simply because it's adjacent to other base pairs that do have more events, more covariations. But these other two examples, they occurred many, many times throughout the evolution of the rRNA, and so we're more confident that they are true. And when the crystal structure came out, all of these were in the crystal structure, including the UUCC. Okay, so this is what I had said earlier. The accuracy of the comparative model was about 97% of the base pairs that we predicted in the 16S were in the crystal structure. About 98% of the base pairs predicted in the 23S were in the crystal structure. 16S is on the left, 23S is on the right. The 23S is about twice the size. It's about 3,000 nucleotides in length. Okay, next part. Current tools and the CRW site. So, I've already told you, I don't program. Although I know Unix, I'm not sure if that's a bad thing to say in here, but we actually did all this work on Sun Microsystems computers from the very beginning. And I worked with programmers to develop a number of programs. A sequence alignment editor. This is essentially just a manual, a system where someone, a human, a mere mortal, would just essentially insert gaps, dashes, to align sequences. And so I spent many, many years using AE2 to align sequences. The program query analyzes these alignments, and we do our covariation analysis. XRNA is the program that we use to help draw these secondary structure diagrams. What we call the RDBMS is simply a simple MySQL database system that we set up like 15 years ago just to keep the inventory of all the data, because the number of RNA sequences is extremely large. I'll talk about that a little later, but I'll say now there's over 500,000 rRNA sequences. There's more rRNA sequences than there are for any other gene, and the reason is because it is used to determine the phylogenetic relationships. It's the best molecule to determine the phylogenetic relationships. So as a byproduct, I get access to all this data that people are sequencing these to determine phylogenetic relationships, and I use it to actually work on structure. So it's been very nice, but we set up a simple system that kept an inventory of all of our data. We have developed a toolkit called CAD, Comparative Analysis Toolkit. Its objective is to essentially facilitate the alignment of sequences and to do many of the operations that I used to do manually. I'm just touching on these issues very, very quickly. CRW is the name of a website that we created. Stands for Comparative RNA Web. And its objective was that in the process of doing all this comparative analysis to predict RNA structure, we were creating a lot of alignments and secondary structure diagrams that the general scientific scientists wanted. And they asked me for it. So we created this website that's called CRW. It is not coincidental that Carl Woese, who was one of my mentors, his middle initial is R, and so it's also stands for Carl Woese. It's probably too small to completely appreciate, but these are a few snapshots of the CRW site. We have recently updated it. This is the newer version. This is the home page. And here we would have a list of all the available 16S sequences and all the 16S secondary structure diagrams. And so we generate secondary structure diagrams for many of the sequences. And this is a picture of an alignment. And this is still a small alignment. We have alignments now that have over 80,000 sequences in them. That's 80,000 in the Y direction. They can have 3,000 nucleotides in the X direction, but in terms of columns, you can have 10,000 columns. So it's a big, big data set. We have various pieces of information like nucleotide frequencies, base pair frequencies for every base pair in our predicted structure. Much of this information, or some of this information, was created statically. Some of it comes from our MySQL database system. It's a bit cumbersome. We created the system called RNA Map. This is a Java system. And our objective is to begin to bring all this information together so that normal scientists that want to learn more about rRNA or any RNA, and they don't want to spend all the time that I spent putting things together, we created this Java applet that in this case has a 23S rRNA, and that this will link to various pieces of information. So what I did here, which I thought was pretty cool, was that we actually read in the 3D crystal structure. And as you mouse over a region, those nucleotides that are within a certain distance of the nucleotide that you've moused over will show up in red. And so red means that you're very close together in 3D space, and then yellow means that you're a little further away, and green means that you're even further distance, and no color means that you're really far apart. It's just a little useful tool for you to begin to transform this secondary structure into a 3D structure, meaning that this region of the 23S is in proximity with this region and this region.
So, Robert, can I just ask you a question about that? So what we're seeing here, the mouse is, when you took the screenshot, the mouse was hovering over a nucleotide.
Yeah, and I forgot which one. Oh, it might be this one right there.
Okay, and given that the mouse was hovering over that nucleotide, what we're seeing is the redder the other nucleotides are, the closer they are to where that mouse is.
Yes.
So if I moved it around, the
yes.
change.
Yes. Okay. Yes. What we actually wanted to do is to make a movie. We started doing it, but it was too cumbersome, and so we need to improve how we do it, in which you would automatically, the mouse would transverse over every nucleotide, and essentially that would become a movie. And so you could see colors changing, and okay, and this is probably too small to appreciate, but here's our RNA map in the center, and it's to show that there's different RNA structure motifs, which I'm not talking about, but I've mentioned that there's base pairs. Base pairs arrange themselves into a standard helix, but they also arrange themselves into these different structure motifs, which I'm not going into any detail, but we and other groups are identifying these. They have actually unique 3D structures. They are involved in tertiary interactions, protein binding. So we are collecting a lot of information about the basic building blocks of RNA structure. So the idea here again is that with the RNA map, you can begin to access different pieces of information. Base pairs themselves arrange themselves in different ways to give you a different conformation. You always think of just Watson-Crick AU base pairs form. Well, there's 14 different ways that the two AU base pairs can arrange themselves, first of all. And we have actually determined the conformation of every base pair. And you also get the non-canonical base pairs, as I mentioned before. You can get UUs and CCs, etc. And all of those have 14 or 15 different ways of arranging themselves. So our objectives are to actually learn at a chemical, at a very fundamental level, the basic building blocks of RNA structure. And ultimately, our goal is to be able to predict the secondary and tertiary structure. And I believe that knowledge of all this type of information that I'm showing here is going to be important. But currently, all this information are in flat files all over the place. And there are people that said, well, I can just write some Perl scripts and I could link everything together. And I I'll mention it, I'll get to it. I feel very fortunate that we were able to meet Stuart, and essentially what we will be doing is bringing all this information together. So when I say from past to future, lessons from, lessons from evolving RNAs, problem effectively using large volumes of information, spanning several different dimensions being sequence, alignment, secondary structure, structure motifs, 3D structure information, phylogeny, base pair conformation, anything, any dimension that is essentially related to this RNA molecule. Okay. Here's the quote that Carl Woese has in a paper. I'm an author, and I jokingly wanted to say et al., Guttel et al., meaning that, yes, I'm an author on this paper, but Carl wrote this. Okay, so what did he say? The comparative approach indicates far more than the mere existence of a secondary structure or element. It ultimately provides detailed rules for constructing the functional form of each helix. Such rules are a transformation of the detailed physical relationships of a helix and perhaps even reflection of its detailed energetics as well. One might envision a future time when comparative sequencing provides energetic measurements too subtle for physical chemical measurements to determine. This statement is absolutely amazing because it was made in 1983. In 1983, many scientists, experimental scientists, were laughing at us saying you cannot predict a secondary structure just by looking at sequence. And here Carl's saying, oh, well, of course you can predict the secondary structure, but here he has the audacity, which is typical vintage Carl, to be saying it's going to tell us a lot more. What he is saying here is absolutely true. I will not be able to go into it in any detail, but we are actually using comparative sequence information, the frequency of certain types of structural elements, and we are finding that there is a direct correlation with the frequency of certain structural elements and their physical stability. So Carl's absolutely correct. And we need the SQL-server system to actually achieve that goal that I just mentioned, or goal, that project that we're working on. And then, I don't know if I should read this because I'm running a little behind, but I'll still do it. We can imagine the evolution of an RNA sequence and structure as a movie, with each rRNA sequence structure a single frame or snapshot. Only a few rRNA sequences were known in 1980, and to a first approximation, their distance from one another on a phylogenetic tree or plotted on an abstract sequence space, such as a principal component analysis, is very large. Accordingly, the amount of variation between each snapshot is significant. However, as the number of RNA sequences increases, this distance between any two snapshots on average decreases. At this time, we have a dense sampling of RNA sequences on some regions of the sequence space graph, which will allow us to map mutations that occur within a small window of time. Our movie of the evolution of rRNA is now beginning to have greater fluency from one frame to another. More detailed steps and pathways in the evolution of large complex RNAs can now be reconstructed. So by having as many sequences as we have, that we can properly, accurately predict their secondary structure, and organizing the information properly, we can now study models of evolution in a way that no one else has been able to do. How much data do we have? This is May, so this is already way out of date. But But we study 16S, 23S, 5S RNAs. There's some introns, group 1, group 2, tRNA. There's over 1 million sequences that we have in our data collection at this time. The number of sequences that we've aligned is actually a lot less than the number of raw sequences. And so one of the rate limiting steps right now is aligning these sequences, these raw sequences into a alignment. And this number just shows that we have 33 different alignments that contain all of these raw sequences. And then we are generating these comparative secondary structure models for a subset of these. And so a model means it's a drawing, and structure information means it's not a drawing, but it's a file that's telling you which positions are base paired with each other. Our goal is to actually have structure information, base pairing information for every single sequence here. This is very doable if we actually develop the right tools, or if I could get a few thousand monks to work at the computer. Okay, three-dimensional structure. So let me back up. We want to now put all of this information into a database system. Currently, we just have the annotation. We just say that we know the names of these 540,000 sequences. And we know it's a 16S or a 23S. And we know how many nucleotides it is. And we know where it is on the phylogenetic tree. We want the sequence in the database. We want the alignments in the database. We want the structure, the base pairing information in a database. My mentors used to tell me, Robin, no, it's not possible. And I'm manic in that sense. Unfortunately, people like Stuart allow me to be manic because he's helped essentially allow us to implement this, which is that we want to put all that information into a database. We want to put the 3D coordinate information for the 50S ribosomal subunit. That includes the 23S and 5S, and the 30S ribosomal subunit. That includes the 16S, into the SQLServer system and do analysis of this 3D coordinates in the SQLServer database system. This is doable. We also want to put the entire phylogenetic tree into the system so that every sequence is mapped somewhere onto this tree so we can determine essentially the evolution of every nucleotide through the evolution of the rRNA. So this is just a diagram showing that here we have an alignment that is currently in the SQLServer system that Stuart developed. That's called rCAD. And we have the tree of life. We have 3D structure. We have secondary structures all in. Okay, this is the quote that Stuart wrote, or this is part of a letter that Stuart wrote for a grant proposal that I submitted in March of this year. Our collaboration began in February of last year when you and your graduate student Kishore approached Microsoft with an extremely complex database problem, how to best represent large-scale metadata, sequence alignment, et cetera, et cetera. The challenge and complexity of this problem were music to our ears at Microsoft. I recently moved to Jim's group after spending 5 years on a team that engineered SQLServer database product and was eager to tackle. I will say, I don't know if I've ever told Stuart this, but Kishore and I actually did not think that this was doable. But we thought, heck, if someone wanted it, I'm not paying the person. If he wants to try it, great. I wouldn't be here if the system wasn't working. So I'll say that I'm here because Stuart was able to help work with my student Kishore and to get the system to work. Sort of an overview.
Kishore didn't think it was. If I knew Kishore didn't think it was possible, I wouldn't have even tried. So that's funny.
He made the first contact with you, and he said, "There's this guy that says that it's doable," and I just don't think so. But because you kept after him, meaning if you didn't keep after him, he probably would have just dropped it. But and after about 2 weeks, he was very excited about it. So we essentially want to develop principles of RNA structure, which is a general concept, and that it's going to need these various components, of which we need phylogenetic information and RNA structure information and knowledge base information, which is knowledge that we learn from our alignments and our phylogeny, and we need the proper hardware and software. Okay, part 4, of which I said I would go to 220, and I still got another section to go, but I'll try to go quick. Future development. The project with Stuart, Stuart called rCAD, for RNA Comparative Analysis Database. But at the meeting I went to in October in North Carolina, the Microsoft meeting, several people said, "But Robin, why don't you consider a TCI application in which you actually can do more?" And I thought, I can actually fantasize about things that we really want to do. What we really want to do is, rCAD is extremely useful because it has all this information that I just discussed. The problem with it is that you need to be a programmer like Kishore or Stuart and write SQL commands to essentially get hands on the data. So what we want to do is to develop visualization or interaction tools that allow you to align the sequences, to see the alignments that sit over rCAD, that allow you to generate these secondary structure diagrams. And so that's the xRNA program. To do the covariation analysis. We also need CAT. CAT was Kishore's thesis, Comparative Analysis Toolkit. This is a tool that that helps align the sequences into their proper form to generate secondary structure information. Currently, much of this was a very manual effort. Again, either we get a few thousand monks or we improve upon CAT. You might say, why do you still want to do it? This is CAT 1. CAT 1 allowed us to scale up, but we still need to make many improvements to essentially align all the sequences. So the development tools, rCAD. There's still more that we want to do in rCAD. We still want to improve upon CAT. We want to develop these visualization tools. So we will need, because this is going to get to be a pretty complex system, we'll need some HPC load balancing, but at lunch, Stuart said that's something that we should be thinking about in a few years. That's fine, but I think it's still going to be needed at some point. CRW services. My CRW site currently is static. All the data was statically created. You go and you get it. We want to make it dynamic, that you can actually go and do things dynamically with the data. Sophisticated data analysis. We want to now analyze data in the rCAD system. So I and Stuart will give some examples of what we are doing here, in which we're now analyzing data in the rCAD system. Okay, managing. I forgot what that slide was about. Oh, I think this is. Okay, this is actually something. Yeah, it's something that you'll cover. We want to improve RNA map. So I showed it before, and I gave a few examples. But we want to be able to click on a region and actually get a 3D viewer in which you can see the actual chemical structure. You'd want to highlight a region here on the secondary structure and see it highlighted on its 3D structure. You want to get base pair nucleotide frequency information from simply clicking on nucleotides here. We have part of this working right now. This was a mock-up in Kishore, my graduate student's thesis, of something that we would want. An alignment editor. You see a secondary structure browser. You see a phylogeny browser. You can click on a part of the structure browser and it would bring you to the appropriate region alignment or vice versa. And the same is true with the tree. You click on a branch, a node on the tree, and it would bring you to those sequences in the alignment. This is something that NCBI has put together. It's called NCBI Fast, which stands for Vector Alignment Search Tool. This was developed for proteins. They have a 3D viewer of a protein, and they have different types of structure motifs that are shown here. And they have a sequence alignment of a few sequences for this protein. You can highlight or click on regions here, and the appropriate amino acids, nucleotides will highlight, show here, vice versa. It's an interactive tool. It's pretty neat, but I believe we can do much better, and they are not using the SQL-server system. And not to pound on David Lippman too much, he's the director of NCBI, but I told him about the system that Stuart was putting together, about bringing all this information together, and he said that he doesn't think it would work. And so I would actually like I would like to visit David Lippman with Stuart, if Stuart's agreeable, show it to him, and actually ask him if they might want to do the same thing with their protein system. Okay, analysis and applications. I'm going over, I apologize. Let me talk about two areas. Stuart's going to cover some of the covariation analysis, but we have some slides that we just did that Stuart hasn't seen that I'd like to show. And we're doing structural statistics. Structural statistics meaning is that we can now pull out specific types of information, and I'll give you some examples. Okay, in the covariation analysis, when we say event, it's what I was referring to before. It's a change somewhere on the phylogenetic tree. What Stuart did was, by putting in the phylogenetic tree into rCAD, we can determine how many times a nucleotide or a column or two columns can change simultaneously. And we call that an event. And so what we're showing here is, let me see the next diagram. Yeah, let me show this first, is that all of the base pairs that are colored are those that were identified with the system that Stuart developed. And the darker the color, the more events, the more times it changed throughout the evolution of that molecule. And what you don't see is that we're able to pick up several other constraints that because it's the way that this is drawn, you actually get a lot of crosses right here, which means that what it's showing is that many nucleotides in this helix are changing in a coordinated fashion. It's not just each of the two nucleotides that are base paired that change simultaneously or are constrained, but that many base pairs can be constrained, but that it's a more subtle signal, and you need the phylogenetic tree to actually sniff out that type of information. So back to this, what was shown here is with the threshold, we didn't say it here, was with a threshold of 100, which meant that there had to be at least 100 changes somewhere on the tree, and that about 72% of those mapped to a known base pair, and 28% did not map to a known base pair. And unfortunately, there are ones that are local inside a helix, like right here, or the crosses here that you don't see. But the point is that, sorry for sort of jumping back and forth, jumping too much back and forth, but that I would say the vast majority of this 28% in red here are actually of biological significance that we were able to sniff out by using the phylogenetic tree information. This alignment, this analysis that we did, was a prototype. We wanted to get a larger alignment before we actually brought it to publication. But then I found the paper, and my PhD advisor's name on it, and David Hostler at Santa Cruz, in which they were doing something similar. And they said, we found all these new base pairs by using the phylogenetic event method. And so my heart was thumping, and I was all nervous, and I started looking through their paper, and I just thought, I had real mixed thoughts. First, they were picking up a lot of, or they were predicting a number of interactions that were not in the crystal structure. We only pick up one. I didn't say that before. This one yellow right here is the only false positive. They picked up like 30. And they claim they found about 25 new interactions. And it so happened that I had already found them and published them, and some of them 15 years ago. And I had to send them a very polite letter with attachments of each of my papers highlighting that I found them. It's not to be too harsh on them, but I was sort of shocked that could be published, that they claim they found all these new things when in fact they were found a long time ago. And therefore, my reason for saying that is that if that can be published, if we want, we could publish this version right here. Even though we wanted to bring in more alignments and do more stuff. Okay. 2 more slides. Structural statistics. With our system, you can now snap your finger. Well, it's easy for me. I snap my finger, and I could, which is essentially telling my graduate student to go do something. And sometimes in a day, sometimes an hour, sometimes a week, he comes back and he gives me some information. What we wanted to do here was just ask, what is the actual distribution of ACGUs in the paired or helix and in the loop regions? And the greens, so for example, 30% of the adenines are in a helix. And 70% of the adenines are in a loop or a unpaired region. In contrast, CGs and Us have many more nucleotides in a helix and a lower percentage of nucleotides in the loops. And so the ratio of loop divided by helix, you see that over like almost 2.5 A's are in a loop region versus less than or more CGs and U's are in a helix versus in a loop region. We now begin to understand this. In the 3D structure, they started finding that adenines are involved in many, many tertiary interactions. So even though we didn't understand why there was this bias, we can now begin to appreciate it with the 3D structure. And then I just wanted to emphasize that when you look at the 3' end of a loop, just at the very last 3' end, 50% of all the 3' end of loops is an A. That's an extremely strong bias that we don't have an explanation for. But it's just to give you one of many examples of a structural statistic that we can pull out of our system. And yeah, and so this is the last one. And this one is really cool, so I'll try to explain it. What my student David did is he would take an entire sequence and look for a sequence of length 4 and then look for 2 closing base pairs that flank it. And so unfortunately I didn't draw on this to explain what we're doing. But we're essentially looking for a UUCG. Every time you find a UUCG, you would look for, say, a U here and a corresponding A here to base pair, and a G here and a C here. Does that make sense, what I'm saying? Or that essentially you
So candidate base pair capping the
yes.
at the end.
Yes. So when you find a UUCG sequence, there's over 30,000 examples of those that are closed or flanked with a Watson-Crick base pair, with 2 or more Watson-Crick base pairs. But 27,000 of those are actually in the comparative model. So as a predictor, 88%, if you were a gambling type person and you wanted to start to develop an RNA folding algorithm and you found a UUCG that was closed by 2 or more Watson-Crick base pairs, you would say, I'm going to put that in a secondary structure model. And then we show a few other loop sequences and their percentages. So for example, GCAA, closed by 2 or more Watson-Crick base pairs, about 53% of those are actually in the comparative model. We can go further. You can now start to ask, what type of closing base pairs is it? It might be that when you have a GC that actually closes or closes this GCAA base pair, this ratio might even go up higher. This is just sort of a first round. There's 256 sequences of length 4. We skipped 246 of them, and now I show the last 5. So here, these 5 sequences of length 4 that are closed by 2 or more Watson-Crick base pairs, they occur by chance, but none of them are found in the comparative structure. Okay, I'm running behind. I get some funding from NIH. Hopefully they'll continue to fund me. I've received some funding from Microsoft Research, which is greatly, greatly appreciated, and the Welsh Foundation gives me some money too. This is an accurate list. We have, I don't know, 8 or so different publications or papers that we want to write. Stuart's going to be an author on all of these. I actually have about 5 more papers that I'm working on. Sorry, Stuart, that you won't be an author on. But there's a lot of work that Stuart's been involved in that is publishable, that we want to get published at some point. Future directions, we want to just get more data into the system. We want to develop more types of structural statistics. I didn't show it, but my student David Gardner actually has about 10 different types of structural statistics that he is pulling out, a lot more than what I showed you. But on the other hand, there's still a lot more that I would like to develop. We want to completely integrate the CRW site with the SQL server, as currently it's mostly getting its data, its annotation data from our MySQL system. So in the process of what we call the CRW migration, and so my person who's a MySQL person is learning SQL Server and is in the process of making this transition. We want to develop, as I mentioned earlier, these web services, dynamic access to this information. For the machine learning type people, we have this project that we are in the process of writing a grant proposal for. It's me and 3 other people, in which we essentially want to obtain the structural statistics, so that's doable. And for the people that are more chemical, you can translate these structural statistics into statistical potentials, and there's these 2 professors that know how to do it, Peng-Yu Ren and Ron Elber. And alignment model, Ron Elber also can use structural statistics to improve the alignments. And then this person, Risto, I don't know how to pronounce his last name, is a CS person who does machine learning. So he wants to essentially independently use the structural statistics to try to come up with some information that ultimately would be used in a program in which we can feed in a sequence and predict its secondary and tertiary structure. And one last thing, part of this is out of date, but I just wanted to mention that NSF has this DataNet announcement in which they want to fund like $25 million a year, these large database farms that essentially would be putting out information like what's at the CRW site. And Jay Bousseau, who's director of the Texas Advanced Computing Center, TAC, wants to use MyLab's CRW site as a prototype for this DataNet. So he essentially would want to use the SQL Server system as well. And this is a few places where I've been, and these are all my license plates. And that's it. I'm sorry, I went a bit over.
I'll say I want to spend 2 minutes to give people in the room who have worked with databases before, maybe know some SQL, a flavor for how we approached integrating this information into the relational database. And so let me skip to some of the meat. On the right here is the legacy system that the Gattell Lab had been using, very much focused on a lot of flat files, the sequences themselves, the alignments, sometimes containing as many as 60 to 80,000 large molecules. We're talking about molecules that are 3,000 to 5,000 nucleotides long. In some cases, just loading these alignment files into a computer system to do an ad hoc analysis, to perform a visualization would take 10 or 15 minutes. The beauty, of course, of keeping all of this in a single integrated database system is that it's available on demand whenever you need it. It's in memory. It's at least accessible to memory almost immediately. So the new architecture, which is we have maybe about 80,000, approaching 100,000 sequences inside SQL Server in alignments, and of course there's a lot more to do. The beauty of this system is that it includes all the sequence metadata, the phylogenetic information, the sequences themselves, and their relationships, their positional relationships within the alignments, along with the other annotations, the structural annotations, the diagrams, and the crystal structure data. This project, from my perspective, was a poster child for the capabilities that were new in SQL 2005. Basically, this project couldn't have been done in an earlier version of SQL-server because we leveraged so many of the capabilities that were newly introduced. In particular, the tree structure representing the tree of life, the phylogenetic parent-child relationships, are represented using common table expressions. If you've worked with SQL 2005, you know what I mean. It allows you to drill down into a tree and to do that recursively. We make extensive use of the CLR, both functions and stored procedures, to implement the comparative sequence algorithms, and also to implement the algorithms that let us navigate down the phylogenetic tree and detect changes in sequences at different points down that tree. That's all occurring as stored procedures in memory, but we're using that tree structure in memory to implement these counting algorithms. Other highlights, most of the material, the information that comes from GenBank comes in a feed daily in XML format, and we're using the enhanced XML capabilities in SQL-server to handle that, as well as a number of the data mining and reporting capabilities. A brief look at the schema in SQL-server just to highlight the kind of information we're keeping in the system. The parent-child relationships representing taxonomy, the tree of life, these are the green tables here. The information about the lineage of the individual sequences are in the sequence metadata tables in blue here. And then the sequences themselves are really information that are retained in the yellow tables, along with the relationship of those sequences to the positions in the alignment. The orange tables represent the structure, the secondary structure. And the real innovation, I think, that we took with this project, because you have to look at columns independently, because you have to look sometimes at a subset of sequences and a subset of columns, we split the sequences apart, and in the database, we actually have a row in the database for every nucleotide in every sequence. Traditionally, in relational databases, when you store sequence data, you store long strings. And you pull out a whole string into memory, and you write some algorithms, and you put it back. But here, we're using the database itself to represent that matrix at a very fine grain. And we use some clever indexing so that it's fast to access an entire column, it's fast to access an entire row, and it's, in fact, fast to access a little square as a viewing window within the alignment. Robin mentioned the elements of secondary structure. There are helices. There are different types of loops, hairpin loops, internal loops, bulges, where you have a mismatch. You have a bulge on one end and a helix on the opposing end. All of those are represented in tabular form. And I won't go into great detail, but basically for every piece of an internal structure at a particular location of a particular molecule, we have a starting position and an ending position. So basically we do these neat between joins between locations in a molecule and their structure annotations. So we can ask questions like, show me all the bulges that fall in this particular quadrant of the molecule's structure, and show me their composition. Or, when I see this pattern in terms of sequence, what structures are they associated with? And that's how Robin's team was able to pull out those very interesting and very new conclusions about the composition of different internal structures. I'm going to skip the demo right now because there are a couple of important points I want to make about the scientific workflow at work here. The legacy approach to doing this covariation analysis, the statistical work to deduce what positions in the molecule are really related, really looked like, load these massive amounts of flat file information into 20, 30, 40 gigs of memory on a big workstation or a big server. Run through a bunch of C++ code, if you're lucky C++, maybe if you're not lucky Perl, and then create a bunch of large output files for each run, write a bunch of scripts to parse the output files. I mean, this is typical scientific workflow. Just some metrics to work on a small alignment involving about 4,000 of these sequences. The sort of the state of the art a year and a half ago was to really spend 16 hours. To extract those covariations. And the new approach within SQLServer that's been working really well for the last year, all the data stays in the database. It's always available. We use CLR stored procedures to perform the calculations close to the data, extracting what you need when you need it. You don't need a huge memory footprint anymore. You can get by on a server with 4 or 8 gig of memory. The output results go to a table that themselves can be queried. So you've retained your analysis and your results in a structured form. And using the brute force techniques that were implemented in C++ and Perl, it takes less than an hour to get that same 16-hour result. And using some clever heuristics, we can take another order of magnitude of time down and get it down to 10 minutes. So this is almost interactive covariation analysis. And the importance of that is that as you import a whole bunch of new sequences, and Robin explained he's got a backlog of nearly 500,000 sequences that have yet to be aligned, structurally aligned, you want to go back and iterate. You want to run covariation analysis again. You want to say, what's the quality of this new data that I've aligned? Do I have to tweak it? It's inherently an iterative process. And if you can get that cycle down to the order of 10 minutes, 20 minutes across tens of thousands or hundreds of thousands of sequences, your capability to more fully automate the structural alignment process goes way up. So we used a number of heuristics and tricks to reduce the search space of candidate columns that might be paired. And while we were experimenting with these techniques, we were able to extract the data quickly along with the various key metrics into visualization tools to validate our approach. But the productivity we were able to achieve working with these tools, I mean, the one caveat is that you need to know SQL to make effective use of this. And as Robin, I think, correctly expressed, what you really want is a semantic layer on top of the SQL that translates structural queries, that translates covariation requests into underlying SQL statements, but allows the scientists to be able to get the work done using scientific terms. The last thing I want to show briefly is just a before and after picture of how structural statistics used to be done compared to how they're done today. A limited set of structural statistics were available, 3-4 years ago on your site, exploring just the tip of the iceberg of the types of combinations that were possible. And in order to get this data out, it really required first a big parser of this alignment file to run, 14 pages of C++, and then a Perl aggregation script taking the results of these. I mean, just hard-coded, custom-coded. You really had needed to have a lot of internal knowledge about the data structures in order to get the work out, and it required about a week's time to run, and it produced a comprehensive set of statistics. What we've changed to is a model where you can actually write these compact little SQL statements. It's not a perfect world. It's still SQL, and there's still this impedance mismatch between asking a structural question and turning it into joins in the table. But they're compact, they're concise, they run in about 30 seconds or less, and you can build a large collection of these to run periodically. They're much simpler to manage and much simpler to understand and extend. And in fact, Robin was referring to graduate student David Gardner, who's building an extensive library of these routines to ask these really deep questions about what types of sequence patterns are associated with different shapes, different segments, different motifs that we see structurally in the molecule. And it's just a much cleaner and more efficient approach. Rather than waiting a week to get a well-defined batch of output, to be able to ask these iterative and follow-up questions more or less interactively. So that's really what I wanted to emphasize. We have a lot of other interesting work in this area that's being enabled. I think Robin's team, you've begun to really look for people that have knowledge and experience working with databases and with SQL now to be key parts of your team rather than just Perl scripts and C++ writers. And I think you've seen the productivity of the whole lab really increase, and the nature of the scientific questions that you're able to answer really get raised to a new level. So that's all I wanted to say, a few remarks. I hope to maybe do a longer talk about this from a database-centric standpoint sometime over the next couple of months in the SQL-server building to get more of the database crew over, and I'll be sure to send an email to the bioinformatics discussion group or interest group so that those of you who'd like to attend that can come as well. But I want to pause now in case you have follow-up questions, either for myself or for Robin. Yes?
So just the first time I've heard of somebody really going ahead and not storing those sequences simply as a single element inside the database and really breaking them up, and in your case by nucleotide. A lot of the sequence alignment algorithms are using these dynamic programming approaches that really would be greatly benefited by not having to load both of them into memory, and then on top of it, even though, I mean, they're clearly these first order models, so you don't really need everything there at once.
Mm-hmm.
Have you guys played around with the ability to perform some of these other algorithms that are very common in this field in this new approach? And we're not going to just load a whole sequence into memory both times before we start working on it.
You know, I personally experiment. I've built sequence alignments algorithms, Smith-Waterman and you know, Needleman-Wunsch in C#. And I haven't brought them into the database, but it's trivial to bring them into the database because SQL-server supports C# running stored procedures now. So I would say conceptually I've proven that it can be done. The challenge is that most of the reference implementations of these algorithms, like FastA, like any of the core reference implementations, have not yet been ported to the CLR, to our stack. So to get, to write something that's publishable saying, I'm using the FastA for this alignment, you still have to get it out of the database into some in-memory venue and bring the results back. But it's conceptually, there's nothing stopping us from going there. It's just a matter of just time, resources, debugging, making sure that we get the exact same results in all the test cases, and bringing it into an environment where it can actually be a lot closer to the data. But dynamic programming, easy to implement in C# and easy to implement anything in C# inside SQL-server. So there's nothing to stop us from bringing that closer to the data as well. Good. Other questions? Okay, thanks a lot for coming.